Welcome back again, guys. This is finally part 3 of our what is turning into a very long lesson on cell transport. So we're still discussing ways that materials can move across the membrane, move from outside of a cell to the inside, or the opposite. Sometimes the substances are moving from the cytoplasm to the outside of the cell. And so in part 3, we're going to cover concept 5.5. This is found on pages 112 and 113 in the Campbell Biology in Focus textbook. So it's just 2 pages, so maybe I won't be quite so long-winded this time. These last 2 big types of transport that we're going to discuss are sometimes referred to as bulk flow, because instead of individual molecules crossing through a phospholipid bilayer or passing through a channel protein or being carried across kind of one at a time by a carrier protein, this is large amounts of substances being quickly sort of scooped up or gulped or swallowed up, you might say, by the cell, or spat out by the cell. So it's I guess the biggest fundamental difference between this and what we've discussed previously is just sheer quantity. So bulk flow, endocytosis, and exocytosis are when large quantities of materials are moved either in or out of the cell kind of all at once. And so I'm going to start with, I think, the one that's the most intuitive, the easiest to understand. It's called phagocytosis, and it is a type of endocytosis. So let's just break down these root words. Endo meaning we're taking in something. CYT, by now you should know anytime you see CYT in biology, it means something to do with cells. So this is literally cells taking in materials. Phagocytosis literally translates cell eating. So not all cells are capable of phagocytosis. Examples of this, cells that can do this, would be like an amoeba, which is a single-celled protist, a protozoan that lives in pond water, a little microscopic organism, and it doesn't have a mouth, and so it obtains food through this process called phagocytosis. But in your body, excuse me, a great example of this would be some of your white blood cells. They're called macrophage cells, and they have the ability to literally gobble up bacteria and viruses and parasites that are making us sick. And of course, a white blood cell doesn't have a mouth, so the way that it ingests food, keep in mind this food particle, we're not talking about like food that you have eaten that passes through your stomach and your intestines. That's not what this is. In this case, the food particle would be, let's say, a bacteria that's in your body that's causing an infection. And so this large cell would be the white blood cell. And notice it has the ability to kind of change shape and kind of fold out part of its cytoplasm and change the shape of its plasma membrane to create these little, almost leg-like protrusions called pseudopods or pseudopodia. And basically the pseudopodia stretch out and wrap around, as you see here, the bacteria until all of this fuses together, and basically the bacteria is now inside the cell, completely surrounded by membrane, enclosed in what we call a food vacuole. And then very quickly, the lysosomes of the cell will swarm and attach to that food vacuole And you know what lysosomes do. They dump enzymes, very powerful hydrolytic enzymes. They release those enzymes onto the bacteria until it is digested and killed and destroyed and broken down. And then all of the lipids and the proteins that were in that bacteria can now be recycled and used by the blood cell. So this is one of the basic mechanisms by which you fight off infections. It's literally a large cell eating a smaller cell by wrapping pseudopodia around the cell, the small cell, what we're calling a food particle here, until that tiny cell is completely surrounded, completely swallowed up, and then digested. Phagocytosis. Yes, it requires energy. This is food particles being eaten by a cell, but remember, we're not talking about the food that, like a hamburger that you would eat. We're talking about other cells that this large blood cell or amoeba cell is swallowing up. How does the food get swallowed up? By pseudopodia. Those are the little legs that stick out and wrap around. And then there's your description. And feel free to pause this and take notes as needed. So we just talked about phagocytosis. This is just another representation of what it looks like. And remember, phagocytosis falls into a larger category known as endocytosis. Endocytosis meaning large quantities of materials being quickly taken in. So another type of endocytosis is called pinocytosis. Now, if phagocytosis is cell eating, what do you suppose pinocytosis might be? If you guessed cell drinking, you are correct. In this image, the tan area, the beige area, whatever color that is the inside of a cell, the cytoplasm. And then, of course, this would be the plasma membrane shown in gray, phospholipid bilayer. And then the blue area is what we call extracellular fluid. So we've talked about that a few times. That's just the liquid outside of the cell. And that extracellular fluid very often will contain nutrients that have been delivered by a nearby capillary blood vessel. So all these little dots and triangles represent things like glucose, amino acids, various ions. And, a lot of these materials can pass into the cell by simple diffusion or facilitated diffusion, but that takes time. And sometimes the cell needs a large quantity of these materials and doesn't have the patience to wait on the relatively slow process of diffusion or even active transport, which can also be kind of a slow process. So sometimes what cells will do is the surface of the membrane will pull inward. It'll literally fold inward. That's called invagination. The cell membrane folds in, creating a little bit of a pocket. And what that does, it causes some of this liquid to flow into that pocket. And then eventually that pinches off and forms a vesicle, a tiny container with membrane wrapped all the way around it. Basically, the cell has taken a gulp of extracellular fluid. And now that extracellular fluid is now inside the cell, packaged inside this vesicle. And hopefully there'll be nutrients in here that the cell can use. Glucose, amino acids, nutrients. So pinocytosis is literally cell drinking. It's fluids and any nutrients that are dissolved in that fluid being quickly gulped into the cell and trapped inside vesicles. So it's the rapid uptake of extracellular fluid. And this happens very often when cells are located really, really close to a blood vessel, because that's when you're going to have the highest probability of having a high concentration of nutrient in that extracellular fluid. Now, if you look at the next picture down, you see there's this, it's a ridiculously complex name, receptor-mediated endocytosis. It's very similar to regular pinocytosis. In fact, receptor-mediated endocytosis is a type of pinocytosis. And what's happening is basically the same thing. The cell is folding in and fluid is being gulped into the cell and packaged inside of a vesicle. But the difference is this. With receptor-mediated endocytosis, instead of the cell just taking a random gulp of fluid and just hoping there's nutrients present, in this case, the cell uses these little, you see these little Y-shaped proteins? Those are called receptors. They look like the letter Y. Those receptors are specifically shaped so that they will only bind with a particular nutrient. In this case, the little circles will not fit on the receptor, only the little triangles. You see the little purple triangles? When those triangles hit the receptor, it's basically telling the cell, hey, there's some nutrient that's definitely present out here in this fluid. And the cell will take a gulp of fluid only when that nutrient is present. Now, there's probably lots of examples of this, but the best example that I know of is when your body cells take in cholesterol molecules. Cholesterol is a steroid that's needed by cells because cholesterol is kind of the starting point that all other steroids are made from. And so you need to take in cholesterol, not only because it's useful for the cell, but it also gets some of that cholesterol out of your blood. And too much cholesterol in your blood is a bad thing. It causes heart disease. So this is really important. You have receptors on the surface of your body cells, and they are cholesterol receptors. And they just sit there, and they don't do anything until there is some cholesterol present in this fluid. And if there is, as soon as the cholesterol binds to the receptor, it triggers a rapid infolding of the membrane, and the fluid is gulped into the cell, not just the fluid, but also, of course, the cholesterol molecules. And so this is called receptor-mediated endocytosis. This is how cells take in specific nutrients by gulping extracellular fluid only when the receptor is bound with a specific ligand. That's just a fancy word for a specific molecule or nutrient. And the example that I used was cholesterol. So those two processes, pinocytosis and receptor-mediated endocytosis, very, very similar. In both cases, the membrane folds in, causing fluid to be sucked in and trapped. The difference is down here, the gulping is only going to happen when a particular nutrient is present. And it has to physically bind to the receptor to trigger that folding in process. Now, the opposite of endocytosis is exocytosis. And if endocytosis is gulping in large quantities of materials, exocytosis is the exact opposite. It is expelling or getting rid of or dumping out large quantities of material. So let's take a closer look. So a common scenario where exocytosis happens is when your cells have produced some sort of useful protein product. Now let's think back to our cell structure lesson. You know that proteins are built by ribosomes. The ribosomes are the protein factories, and the proteins are very often made on the rough endoplasmic reticulum. And then if that protein needs to be shipped out of the cell, if it needs to travel somewhere else in the body, it's going to have to leave the cell in which it was produced and then travel usually through the bloodstream to some other cell. So what part of the cell is responsible for packaging proteins? Do you remember? That's right. It's the Golgi complex or the Golgi apparatus. And what the Golgi does is it packages the protein. These little red dots represent some protein that's been made by the cell. The Golgi's job is to package that protein in a what's called a secretory vesicle or a Golgi vesicle. It's just a little package. It has phospholipid bilayer membrane around it, and it's just chopped full of some useful protein. And so this whole vesicle will slide out to the edge of the cell. Now, the yellow is the cytoplasm, so this is all inside the cell. The vesicle is going to slide out to the edge of the cell where the plasma membrane is located. And the membrane around the vesicle and the membrane around the cell is made out of the same stuff, right? It's phospholipid bilayer. So when those two touch each other and they come into physical contact, they can sort of smoosh together, they fuse together and become one. So now this membrane is, or the vesicle membrane is now part of the cell membrane. And all the stuff that was inside that vesicle, all that protein now can be dumped out into the extracellular fluid, which is out here. And probably there's going to be a blood vessel, a capillary that's going to pick up that protein and carry it off to some other part of the body. A really good example of this would be a hormone that we've talked a lot about in our class already called insulin. You know, insulin is made by your pancreas. So this cell would be a pancreas cell, and the ribosomes in the pancreas cell would build the insulin. Insulin is a protein. It's a protein hormone. So each red dot is a molecule of insulin. But insulin does not do you any good if it stays in your pancreas. In order to function, it has to leave the pancreas, get into the bloodstream, and travel to all the other parts of the body, especially the liver. So the process of secretion, which is a type of exocytosis, is really important. After insulin is made, it gets packaged by the Golgi, and then eventually the vesicle from the Golgi fuses with the plasma membrane. This is all happening inside the pancreas cell, and then here comes the insulin being dumped out of the pancreas cell, where it will eventually be picked up by the bloodstream and carried throughout the body. This process is called secretion, and it's super important. Yes, it requires energy. All of the bulk flow processes require energy, both endo and exocytosis. But this is where some useful product, usually a protein like insulin, is being secreted or exited from the cell, starting out inside of a vesicle, but then eventually the vesicle fuses with the plasma membrane and the product is literally dumped out to the outside world or to the extracellular environment. Now, exocytosis can take on a slightly different form. If you go back to the image here, let's suppose instead of some protein product, let's suppose that these molecules are simply waste materials that the cell is needing to get rid of. In that case, the process would still be very similar. The Golgi is going to package the waste material inside a vesicle. The vesicle will fuse with the plasma membrane, and the waste material will be dumped out. The only difference is if it's waste material, we don't call it secretion, but rather we call it excretion. It's the same process. It's some material packaged inside of a Golgi vesicle that is then being dumped out of the cell. And that is the end of my lesson on endo and exocytosis. And this one was a little shorter than the others, only about 21 minutes. So I hope I didn't go too fast. If there's anything you didn't quite catch, please, please, please go back and watch it again. And you know there'll be a quiz coming up soon. I hope you have a good rest of the day. Take care.